Good afternoon, everyone. My name is Steven Zabo, and these are my group members, Oase, T, GP, and Hardik. And we are doing a project on the early detection of melanoma via image segmentation. Our objectives for our project were to determine the ratio of the surface lesion in a skin cancer picture to the underlying angiogenesis through the image segmentation. Originally, our hypothesis was the ratio is going to be about 1 for the benign lesion, 1.05 to 1.2 for a dysplastic lesion, and greater than 1.25 for a malignant lesion. And with the way we're going to do this is we're determining which color space is most effective to perform the segmentation. There are several different ones, examples of RGB, grayscale, or the LAB color spaces. And then we're processing the images in those color spaces to figure out which is going to be the best for the segmentation. And the most important objective here is to automate the entire process, specifically so that when you take the pictures of the skin, you're not going to have human interaction to adjust things or anything like that. And why are we doing this? Specifically, each year, more than 50,000 people in the United States alone are diagnosed with melanoma. And of those people, 1 American every hour dies from malignant melanoma. Early detection of this cancer reduces the number of deaths due to the cancer. Our approach to figuring out how to do this is, what we're going to do is we're going to take the 2 different types of pictures and analyze them. We've got surface illumination, which is the surface, and then transillumination, which is showing us the blood flow underneath. We're comparing the area of this, like I was saying before, the surface lesion compared to the angiogenesis, which is the blood flow specifically for that lesion that is supplying the oxygen to it. We are comparing the results of our analysis with the projections, our hypothesis that we were suggesting before. The state-of-the-art things that we're using the Nevoscope, which is a device created by Dr. Adam Dewan. It's a photography device that allows you to take pictures of the lesion and the underlying blood. Also, we're using MATLAB to create the image segmentation results using the image processing center that's included with it.
Just to detail the process we're going to move to do the segmentation, there's many steps that we had to take. Overall, the idea is pretty basic in the fact that we're just comparing an area, but the steps that we have to take to pre-processing is numerous. First off, we have to analyze all of our sample images and also just filter out and just weave out any poor images where they suffer from oversaturation, overexposure, or other various issues such as too much hair. Sometimes we have an image where the lesion is covered by so much hair that you cannot analyze the region correctly. And also, what we also have to do is determine the region of interest, or the ROI, and that is done by looking at the actual image, and when you take an image, the image is actually circular with a black ring around it, so we actually have to cover that and get rid of that for our analysis, else that'll be included in the area of our segmentation. So we compared the different color spaces. We found the best color space to use was the LAB color space. We found that not just looking at the individual spectrums, but also just for manipulation of the different spectrums put together, where we either added or subtracted the color spaces from one another. And to analyze, it shows right here, to analyze the results, we used a combination of the A space plus B space minus the L space for surface images, and then we used the L space multiplied, a dot product, with the A space for transluminant images. And then we compared the area of the surface image to the blood flow region underneath. Okay, and this is just a picture of sample output from our code. If you look here, this is the original image of the surface lesion, and this is actually for the L color space, the A color space, and the B color space. And over here, this is when we do our mapping of the different color spaces together. And then afterwards, after we run our code, this is the segment that we have of the original lesion that we cut out. And this right area is the area that we use to compare the ratios. And then if you look down here, this is the transilluminous image, which is the image of the blood flow underneath. And then this right here, again, we broke it down into different color spaces. And then this is also the dot product of the L color space and the A color space. And this white area shows the area of the angiogenesis underneath, which we use to compare the ratios of. Pseudo size of the images we used, we've had about several hundred images, but due to the actual diagnosis of images and the various issues with them, we were limited to about 10 to 20 pictures for each case. We found a range of ratios found through our sample images to be 1.0177 to 1.0696 for a benign lesion, and then 1.0963 to 1.2471 for dysplastic lesions, and then we found that in all of our malignant cases, it was greater than 1.727 as the ratio. The resulting ranges found conform to the hypothesis pretty well, especially due to the fact that we have a 100% true positive detection rating for malignant analysis. We have a 77.78% for dysplastic due to several outliers, and also 69.23 for benign cases. But we feel that the errors due to the benign and dysplastic is that the ratios are relatively close. So whenever we had outliers, it was hard to tell which was which. Future work that we wish to do is the MATLAB code. It was originally developed by Hardik and GP, and it was debugged by Malik, in which Stephen and we did all the processing of it. And then what's another thing we need to do, which we haven't been able to do, was to automate the actual ROI detection. The region of interest inside the circular ring from those pictures before, we actually have to define manually by hand and tune, and we wish to implement that. We're probably looking to implement that through the analysis of the histograms. And also, not only that, we also have to automate the detection of the threshold levels for determining whether, when we transformed it to the black and white, whether we should count a pixel as black or white. Because with every image is different due to the fact that people have different color skins, due to the tone of the tumor and such. So also, and finally, we also need to create a classifier for the type of lesions. Based on the data we received, it modeled towards our hypothesis pretty well. So we're going to look to we want to use a larger data set, and so we could find a more uniform this place for all the data. And just some references and acknowledgments. I just want to thank Dr. Dewan and Dr. Sachin Padran for giving us all the information. All the previous work was done by, most of it was done by those two people, and it gave us a lot of insight to work on this project. And also, they're the ones who provided us with the images for the Novoscope and also the references in order to help build our segmentation code.